Hello everyone, I'm Chun-Yu Tse. The title of my talk is Concurrent Application of TMS and Optical Imaging, FOS or EROS. My talk is about combining TMS and a type of optical brain imaging methods to investigate causal connection in the brain network. Specifically, we will look at the network underlying automatic change detection. I will introduce what is automatic change detection and the optical imaging methods. The relationship between FOS or EROS are similar to that of EEG and ELP. And at the end, I'll also show you how to apply TMS and optical imaging in studying the causal brain connections. First of all, what is automatic change detection? Let me share my favorite commercial. So,
Visa Pay Wave. For purchases under $100, just wave and go.
Automatic change detection refers to the ability of our brain to detect unexpected events in the environment. This process does not require conscious awareness, or it takes place before the engagement of attention. The detection process may not associate with any behavioral responses. Mismatched brain responses are elicited by the unexpected events. In the EEG or ELP literature, this mismatched brain response is known as the mismatch negativity or the MMN. The mismatched brain signal serves as the error signal to trigger remedial action to direct our attention to deal with the deviant. In the laboratory setting, this phenomenon is typically studied with changes in the physical properties of auditory tones, like presenting rare, high-pitched deviant tones among a stream of low-pitched standard tones. Traditional view on cognitive mechanism suggests the involvement of an auditory sensory memory comparison mechanism. The mismatch response are expected to generate in the auditory or superior temporal cortex (STC) under this hypothesis. However, this perspective fails to explain mismatch response commonly observed in the inferior frontal cortex (IFC) and the ability of the brain to detect abstract changes. Abstract changes refer to the deviation from regular relationship or pattern shared among the standards. The abstract regularity cannot be established by repetition of physical features. In this example, each standard consists of 3 tones, and they have fixed relative pitch differences. The deviants violate this pitch difference pattern. The sensitivity of the brain to abstract changes open up opportunity to utilize the mismatch response signal to study different areas in psychology, like emotion, symbols carrying cultural meaning, or stimuli associated with reward value. The abstract change detection and the mismatch responses observed in the STC and IFC can be explained by the prediction violation hypothesis, which is based on the computation model on predictive coding. Regularity shared among the standards are extracted to establish a prediction model to anticipate future events. The current event is compared with the prediction from the model. When there is a discrepancy, early IFC responses is observed, indicating increase in the model fitting effort. A large enough deviations elicit the STC mismatch responses, which serve as the prediction error signal to trigger the remedial action and model updating to improve the model for future prediction. This model suggests a forward early IFC to STC and a backward STC to late IFC connections. FOS or EROS has been applied to study the functional role of IFC in this frontotemporal network. A generic IFC sensory cortical network in detecting prediction violation has been suggested across sensory modalities. The activation sequence of the IFC sensory cortical network can be reviewed by FOS or EROS. FOS or EROS measures neuronal activities. When a stimulus is presented, let's say visually, at the time 0, 40 to 80 milliseconds later, an EROS response is observed in the occipital cortex. EROS measures the change in the light scattering properties of neuron. When the postsynaptic potential is elicited, there's a change in the membrane potential, then the cell body is swollen. These changes lead to a change in the light scattering properties. The other type of commonly known optical signal is NIRS, near-infrared spectroscopy. It is also known as the slow optical signal measuring the hemodynamic responses. The neuronal activity consumes oxygen and leads to increase in deoxyhemoglobin and the subsequent increase in oxyhemoglobin, which is the flushing phenomenon associated with the both signals in fMRI. NIRS measure the color change of blood associated with the hemoglobin concentration. The superior spatial and temporal localization ability of EROS allow visualization of the activation sequence of a brain network. EROS can be measured by putting an optical source and detectors on the scalp of the head. By using multiple source and detector pairs, the location of the brain responses can be triangulated and reconstructed in 3 dimensions. Here is the EROS mismatch responses elicited during deviance detection in the auditory and the visual modality. Time 0 refers to the onset of the deviance stimuli. In both modalities, they show IFC to the sensory cortex, and then from the sensory cortex, that is the STC or the occipital cortex OC, back to the inferior frontal cortex IFC. The time frame shown in here is consistent with the EEG or the ELP MMM response, which is measured simultaneously. Modulation of the forward and backward connection by absence of deviance, amount of information for establishing the prediction model has been shown. This result reveals the association of a generic brain network with the prediction violation mechanism. However, activation sequence of brain network revealed by any brain imaging method is correlational in nature. We further investigate the causal functional connection from IFC to STC by combining EROS with a brain stimulation methods, transcranial magnetic stimulation (TMS). In TMS, a brief magnetic pulse is applied on the surface of the head to produce a focal electric current to disrupt the functioning of the brain. Due to the optical nature of EROS, we can stimulate and observe the brain response without electromagnetic interference. The superior spatial and temporal localization ability of EROS allow visualization of the TMS effect on the brain network and the investigation of the causal connection. During deviant processing, TMS is applied on IFC in the model fitting stage to see if subsequent STC EROS mismatch response will be abolished. Our result really showed the abolishment of STC mismatch responses specific to IFC TMS, but we observed intact STC responses in the control conditions. These studies establish the causal forward connection from the IFC to STC in processing both the physical and the abstract deviance. The actual design of the TMS EROS study is more complicated as it requires showing the temporal and spatial specificity of the TMS effect. The TMS effect is not produced by auditory environment also, or the auditory confine. Intact mismatch response is also need to be produced due to the prediction violation. That is why the actual design is quite complicated. Here is a result showing that the abolishment of STC mismatch response is specific to the IFC TMS, but observe intact STC responses in the other conditions. Here is the result of other control conditions showing the mismatch response in the STC also. In another study, we also find the IFC TMS abolished the STC mismatch response to abstract changes. In the third study, we adopt a similar idea to investigate the IFC involvement in prediction model establishment this time. TMS was applied on IFC to disrupt the processing of the standard event and observe the subsequent ERP mismatch response to the deviance event. The amount of information available for establishing the prediction model is manipulated. The result shows that TMS on IFC really disrupt the processing of the standard and lead to the abolishment of the ERP mismatch response in the subsequent deviant. Here is more detail about the experimental design. The amount of information available for establishing the prediction model was manipulated by changing the length of the standard train and also number of standard that is paired with the TMS. We compare the IFC TMS effect on the ERP mismatch response across different conditions. The results show that IFC is critical for MMN to a deviant perceived with a short standard train when the information for establishing the prediction model is limited. That is how we would know that IFC probably be involved in the model establishment process. In summary, I've shown some examples combining TMS and optical imaging, FOS EROS, to study causal connections of the frontotemporal brain network in automatic change detection. TMS was applied on IFC to disrupt the prediction model fitting or the model establishment processing, while brain imaging method was applied to visualize the TMS effects, the abolishment of subsequent mismatch response. Our studies demonstrate two expected functional roles of IFC suggested by the prediction violation hypothesis. Thank you everyone involved in this project. Please let me know if you have any questions, or you can contact me directly. This is the end of my presentation. Thank you.